So today I'm going to talk about multistability and bifurcations in epileptogenic neural circuits, which I think is a perfect example of how we can apply the tools and the techniques and the terminology of complex systems to a really important biological and health-related problem, which is the study of epilepsy. By means of a quick outline, I'll give you a little bit of the biological background on epilepsy and seizure, as well as a little bit of the history of how complex systems has been used in their study. I'm then going to walk you through two recent works that we've published. One where we sort of use the tools and technique, or more of the language of complex systems to describe what we observe in computational simulations. And then the other where we actually get more into the guts of the mathematics and use more complex tools like stability analysis and bifurcation theory and things like that. And then finally, at the end, I'll try to sort of foreshadow a little bit some of our exciting current and ongoing work, which we're particularly excited about. So first off, I should explain to you why I'm so interested in epilepsy. And it's quite simply because it's the most common serious neurological disorder in the world. If you're not familiar, epilepsy is characterized by recurrent seizure, which is a brain state typified by hypersynchronous, hyperactive neuronal firing. This is often classified using scalp EEG recordings. If you've ever had a sleep study done and you have all those lovely sort of stickers placed on your head, this is essentially how seizure is often diagnosed. You can sort of see how there's a sort of quiescent brain activity in the first half of this plot. And then afterwards, you see these very high amplitude oscillations that seem to be somewhat synchronized across all of these different electrodes, representing different large scale regions of the brain, depending on where those electrodes are placed. Now, one thing that I found particularly interesting as a wannabe history buff is that epilepsy has actually been studied in some form or fashion going back to essentially the dawn of modern medicine in the ancient Greeks and Hippocrates. And in part because of that, there have been fairly effective pharmacological treatments for patients with epilepsy going back for about a century. These are drugs that act to prevent seizure in patients with epilepsy, because again, the disease is characterized by this recurrent seizure. Now, when we look at these drugs in a little bit more detail, one thing sort of jumps out, which is the fact that despite ongoing drug development, there's this relatively hard ceiling of efficacy. All of these drugs seem to only be effective for about 2 out of 3 patients with epilepsy. And one reason we think this might be is because all of these drugs operate under the same general hypothesis of the disease, that epilepsy is a disease of excess excitation. That's something that makes intuitive sense since I just described seizure to you as a hyperexcitable brain state. An analogy I often like to use is if you think of your brain as a car and seizure as your car careening off at high speeds very suddenly, then it makes sense that that might happen if you give the car too much gas or too little brake. So essentially, all of these drugs act in one of two ways. They either enhance inhibitory signaling via neurotransmitter GABA or decrease excitatory signaling by blocking the neurotransmitter glutamate. And there's also a lot of experimental evidence that goes towards this hypothesis that the excitatory-inhibitory balance is disrupted in epilepsy. But if this were to be all and be end all, if this were the only pathway to the seizure state, we would expect these drugs in some form or fashion to probably be more efficacious than this. The fact that there's this hard ceiling of efficacy seems to imply that there may be alternative, less common pathways to seizure that may be what's occurring in these medically refractory patients, these patients who do not respond to these pharmacological treatments. Now, what I've tried to do in my work is use the tools from complex systems and apply them to understanding these, potentially more complicated, less intuitive pathways to seizure. And one reason why I do that is because there's a really great history of these tools being very useful in the study of epilepsy generally. This might make some sense because I sort of described seizure as this very antithetical state. So physiological brain activity, which is relatively sparse and asynchronous and quiescent, is is essentially the opposite of the hyperactive, hypersynchronous seizure state. And the sudden transition between those two states seems to have some similarities to perhaps a bifurcation. This is strengthened by the reality that any brain can exhibit this transition. Any brain can exhibit a seizure. If, for instance, you're an otherwise healthy person who has a concussion or has a extremely high fever, you might exhibit a sort of one-off seizure as opposed to the recurrent seizure that typifies epilepsy. So again, there are a lot of parallels here between these transitions and perhaps a multi-stable complex system where both this physiological and pathological state are both stable, and what's changing is the sort of the relative basin of attraction between those two states. And there's a sort of seminal work in this field by Victor Gersa and his colleagues back in 2014, where they looked at what a seizure looks like in vitro in a slice of animal brain. I believe these are from rodents. And they sort of classified this based upon what type of mathematical bifurcation might lead to these type of transitions. And again, they sort of took this bifurcation theory, complex systems theory style perspective and said, let's make a model where we have some sort of slow control variable that changes the stability structure of the system and leads to these types of bifurcations and in turn, these type of transitions. This led to what's been called the epileptor. It's a relatively simple system of 5 ordinary differential equations that periodically transitions between the sort of quiescent, healthy brain activity and these period of oscillatory, more active activity that typifies seizure. One thing that's interesting about this model is that whether or not it seizes is fully dependent upon this slow permittivity variable, Z. So essentially, there's a knob that sort of has to be turned somewhat manually in order to trigger that transition into and out of seizure. So that's just a little nugget. I'll sort of get back to that towards the end of my talk and why that's important. Now, as I mentioned, we want to take these tools to try to understand some of these newly proposed, more complicated, more counterintuitive pathways to seizure to see if they're in any way feasible. Just like Caitlin talked about how we can do some things computationally that would be infeasible or intractable and experimentally, this is sort of the approach that we were taking to understanding these complex pathways to seizure. So this first work that we did was motivated by some fantastic experiments done by Michael Chang, who's a grad student in our group. This was done just before I joined. And what he found when he was studying slices of brain tissue that were treated with the convulsant 4-aminopyridine, this is a model for epilepsy, a model for seizure, is that he found he could induce seizure-like activity if he optogenetically stimulated just the inhibitory cells. That's a little bit weird. If I go back to my car analogy, that's like me saying I slammed on the brakes and that caused my car to go careening off at high speeds. If that happened, you might say, oh, there must be something weird going on underneath the hood of my car, some sort of miswired connection that triggered that. It's a much more complicated process. So indeed, we came up with this much more complicated multi-step mechanism by which we hypothesized this might be occurring. I'm not going to go into the details of that. Instead, I'm going to focus just off of this first step. This is essentially the trigger of this mechanism, which we posit is a sudden transition in the dynamics of inhibitory neurons, which leads to a large-scale inhibitory signal that's felt uniformly by a majority of the excitatory cells in the network, leading to the sort of first instance of seizure-like dynamics. Now, this was work done with 2 of my co-supervisors, Francis Skinner, who is a computational neuroscientist, and Dr. Taufik Valiente, who is a clinician scientist, as well as my once and always amazing collaborator, Dr. Humaira Moradi-Chameh. Dr. Moradi-Chameh is super skilled in this very technically challenging experimental technique known as patch clamp recording, where she's able to get very precise electrophysiological data from individual neurons. And one of the things she was able to do using slices of human brain tissue that Dr. Valiente takes during his normal resective brain surgeries is she was able to record from an interneuron, one of these inhibitory cells, both before and after treatment with 4-aminopyridine. This allowed us to get some general characteristics of the neuron's excitability quantified via the FI curve, frequency versus input current. And we in turn generated two models, one of a sort of healthy interneuron and one of an unhealthy, hyperexcitable 4AP treated interneuron. We did this using the Isakiewicz formalism. This is a relatively simple system of two ordinary differential equations, but it's extremely powerful because it mimics a wide range of known neuron behaviors and spiking behaviors. So we fit these models to mimic these important properties of the neural excitability. We then put these models into purely inhibitory networks. So these networks are either all control neurons or all 4AP neurons. And there's only inhibitory signaling. The drive to the network comes from some heterogeneous tonic external drive to each neuron. And we asked ourselves, what's different in this network's dynamics in the control setting versus the 4AP setting? And we actually didn't find anything all that interesting when we just let the network simulate from random initial conditions. When things got interesting is when we actually subjected the networks to a perturbation. This perturbation was a very brief, high amplitude current pulse delivered uniformly throughout the network. You can essentially think about that as instantiating a single instance of near perfect synchrony. The motivation here is that if the stable oscillatory network state is in any way possible, in any way stable, we would reason that sort of perfect synchrony would put us right in the middle of that basin of attraction, and we'd thus be able to stay in that synchronous state. We studied a wide range of these networks with varying amounts of drive and various weights of these inhibitory synapses to sort of get a picture of the whole potential, system. And what we found was that these control networks, the less excitable interneurons, were less likely to exhibit these transitions. More often than not, if I had an asynchronous system prior to this perturbation, the system would very quickly decay back into asynchronous firing after the perturbation. Contrast that with what happens in these 4AP networks. Then, when we give this perturbation, we were very likely to see, or much more likely, to see these networks preserve that synchrony in some sort of synchronous oscillatory state. We ensured this was stable by simulating this for thousands upon thousands of milliseconds and ensuring that this never decayed back into asynchrony. So what this let us conclude is that there is a difference between these control and 4AP networks in their propensity to sort of suddenly transition into a synchronous state. That perturbation that I gave the network was very idealized, but we did some further work in this paper to essentially show that there are more biologically realistic perturbations that could also elicit the same effect. So that potentially explains this first step of this pathway, this trigger. Essentially, someone with epilepsy who may have hyperexcitable interneurons is going to be more vulnerable to the network of interneurons suddenly transitioning into synchronous behavior, which can provide this inhibitory bolus to the rest of the system. Now, as I mentioned earlier, this is a very sort of descriptive study, at least from the perspective of complex systems. I've described what we observed computationally using the terminology of stability and bistability. We didn't actually go in and classify the stability of these systems. That's largely due to the fact that the Isakiewicz model isn't the most amenable to those type of analyses. But we still were able to glean these really important conclusions, again, by applying more of the terminology of complex systems to this study. In this next thing I'm going to talk about, we were able to go a little deeper in the mathematical guts and perform bifurcation analysis and stability analysis and all these cool tools, which you're all widely familiar with. Now, this next study, again, was motivated by experimental data. It was motivated by work driven by Dr. Murati Chameh, where she classified a huge number of neurons from these human brain slices that I mentioned. And she found, somewhat surprisingly, that in these very basic levels of or characterizations of neural excitability, there's very notable heterogeneity in these neurons. This was something that was surprising to us. It was surprising to our reviewers of this paper. It was surprising, in comparison to the rodent literature. Human electrophysiology is a somewhat emerging field in neuroscience, but it's something that when we dug into what literature there is out there, this is preserved across all of these studies. So it's a really interesting feature of these human neurons. We viewed this in concert with a really fantastic theoretical work done by Srijoy Tripathi, who is now a PI who we collaborate with in Toronto. This was done during his postdoc. And what he found in this work was that neural networks with a moderate amount of heterogeneity were better at encoding information from an information theoretic point of view. The third piece of the puzzle is the idea that I've talked mostly about seizure as a state of hyperactivity and hypersynchrony, but it's actually also an information poor state from this information theoretic point of view. So if heterogeneity is the default healthy state, and it serves a role in encoding information, but that information encoding is disrupted during seizure, it stands to reason that maybe that disruption is brought about by a disruption in the physiological levels of heterogeneity between neurons. Lo and behold, that's the case. Otherwise, I wouldn't be talking to you about this hypothesis. And what we were able to do in this study, which is available in bioRxiv, knock on wood, we'll get some good news from the review process relatively soon, is we were able to exploit this, really unique access to human data that we have in our lab to compare regions from the epileptogenic zone, the regions that's clinically classified that produces and initiates the seizure, to cells taken far, far away from this zone and are deemed seemingly normal or seemingly healthy in the human literature. I can take zero credit for these amazing experiments. This is all Dr. Valiente performing these surgeries and Dr. Murati Chameh performing these recordings on these cells. The fact that we can do this on human brain tissue is one of the things I'm so lucky to be a part of this group where, there's only a handful of places in the world doing this. And we're actually the first that was able to characterize these cells directly from this epileptogenic zone, because it's actually very difficult clinically to classify that prior to surgery. And what we found when we compared the sort of disease-affected epileptogenic neurons to our control populations was a significantly reduced amount of variability in this distance to threshold measure of excitability, which was really cool, but it begs the question of whether this reduction in heterogeneity is causal for seizure initiation or is just some epiphenomena brought about by some other effect of epilepsy. This is where computational mathematical study came into play. We recognize that the distance to threshold has a direct analog to a simple shift in the activation function of a very straightforward model of neurons. It's just a Poisson process with a given activation function. These are examples of the sigmoidal activation function here. This term H is just that shift representing the distance to threshold. We created excitatory inhibitory networks with excitatory and inhibitory cells that replicate the levels of heterogeneity we observed experimentally. We also implemented high and low levels of inhibitory heterogeneity, matching physiologically motivated values and the known effects of epilepsy on inhibitory interneurons. What was really nice is that because this Poisson neuron model is sufficiently simple, we're able to perform mean field analysis to simplify it down from a network of 1,000 coupled ODEs, one for each neuron, to 2 ODEs, one representing the mean excitatory activity, one representing the mean inhibitory activity. In this mathematical analysis is where my third supervisor, Dr. Jeremy Lefebvre's expertise as a more mathematically minded neuroscientist was invaluable. Now, what we found is when we first subjected a highly heterogeneous network to a linearly increasing drive, was that the dynamics of the system scaled more or less linearly. The excitatory synchrony, the excitatory activity, and the inhibitory activity all scale linearly with this drive. Lo and behold, when we classified the stability of the mean field system, we found that there was always a single stable oscillatory fixed point. The fact that there aren't multiple fixed points, there's no bifurcation and change in the stability, seems to underlie the idea that there aren't any noticeable sudden changes in the dynamics of the spiking system. Contrast that to what we saw when we had low levels of heterogeneity. We see this very sudden uptick in synchrony that's associated with increases in excitatory and inhibitory firing rates. We said this was analogous to the onset of seizure-like activity, given seizure as a hyperactive, hypersynchronous state. And lo and behold, there's also significant differences in the underlying structure of the mathematical system. This system is multi-stable for low levels of the drive. Two of these fixed points collide in the saddle node bifurcation right around the time in our spiking networks in which we saw this transition. And then it yields an unstable oscillatory fixed point. So the fact that we have this bifurcation showcases why this transition is so sudden. The fact that our fixed point is unstable showcases why we get a limit cycle and we get this oscillation that's persistent that leads to this increased synchrony measure over time. So this allowed us to conclude with some sort of authority that there's an underlying theoretical mathematical principle that showcases why heterogeneity is important. All we changed in this system is the amount of heterogeneity in that distance of threshold, and making that lower was enough to make the system more vulnerable to seizure-like activity. Now, the sort of cherry on top result from this work was that we were able to explain something very surprising from our experimental data using the mathematical theory. We actually found that the neurons taken from the epileptogenic zone were less excitable than their control counterparts, which goes against everything I've said about epilepsy being a disease of hyperexcitability. But what we realize is that heterogeneity can actually explain this result. Heterogeneity is well known to sort of linearize population average activation functions, which is exactly what these FI curves are. You can sort of see that cartoonified here with our simple sigmoidal activation functions, where the more heterogeneous blue population yields a flatter sigmoid that increases the activity at low levels of input, which correspond to roughly what this FI curve characterizes. So we think this is a really cool showcase of sort of the cyclical nature of the interplay between theory and experiment, where the experiment motivated these theoretical computational explorations, and then the theory ended up explaining something surprising experimentally. And really quickly, I just want to showcase what are some of our ongoing work, which is building on this model we use in this heterogeneity work to build a model of seizure that is stochastic. One of the most challenging things about treating epilepsy is that seizures are inherently unpredictable. The idea of seizure prediction is one of the major questions in epilepsy. If you could find a marker that would tell you when someone was going to have a seizure, you could better design some sort of closed loop neurostimulation device to prevent said seizure. Building off of this model that I just talked about, we were able to make a model that transitions into these seizure-like events, not driven by some arbitrary control parameter, but driven by natural fluctuations in the noisy input to the system. This is something that we're still polishing and still getting ready for mass consumption, but we think this is a potentially really cool potential next step in seizure modeling that can try to mirror the unpredictability, the stochastic nature of seizure in patients. So with that, I'll just mention that I'm fairly active on Twitter, so I'd love to interact with anyone who's interested in this. Find me on my website, and thanks to all the funding sources, both directly to me and to my 3 supervisors, which has helped with this work. And hopefully I left some time for some questions.
All right, thank you so much. We do have some time for questions. Looking around. Okay, perfect, because I have a billion. So maybe I missed this, but when you were talking about the bistability in your model and the synchronization of interneurons,
mm-hmm.
you keep talking about seizures, but have you looked at this in terms to interictal spikes that have these bursts of activity in between seizures that we see in the field potential? We have some work out showing that we actually saw interneurons active during those not excitatory cells. And so I'm wondering if you could comment maybe a little on how those might be maybe interictal spikes or a different type of model, not just seizures.
Oh, great minds think alike. This is something that we've thought a lot about in sort of putting forth this this sort of more complicated biological, hypothesis. One of the things that is sort of surprising in the literature to a lot of epilepsy specialists is the fact that these interictal spikes are driven by inhibitory neurons, and sort of showcase that the inhibitory cells are almost more active prior to seizure. And if you think about the role of inhibitory cells being to restrain activity and stop a seizure, that's very counterintuitive. So this is, you're right, this could potentially be something where these instances of interneuronal synchrony represents an interictal spike, and that interictal spike is sort of the trigger for the full seizure-like event once its effects are felt by the system. When I sort of misuse the seizure terminology a little bit too liberally here. When I write papers, I'm much more careful about that than when I'm talking off the cuff. But this, I should clarify that we did not sort of model seizures in this work. And this is, a dynamic that is associated with seizure that we think is preliminary to seizure. It's not seizure in and of itself. All
right, so I'm not going to keep harping on this, but you and I should definitely talk about this because I have much more to say on the idea. But we have a question from Hiroki in the chat that says, very interesting results. Is there any empirical data showing neuronal homogeneity in epileptic patients?
Yeah, so the main data is this recording which we have, which again shows that this excitability measure is significantly, the statistically significance is so important in the experimental world. It's significantly reduced variability in those epileptogenic neurons. This sort of heterogeneity hypothesis is sort of one of the driving forces in the lab right now. And we're sort of in the midst of trying to characterize a wide range of other potential sources of this homogeneity. One thing that we found, which is potentially interesting, is that the particular ionic currents in their expression seem to be more homogeneous in epileptogenic neurons compared to healthy neurons. So there's a wide range of potential sources of this heterogeneity. Another is in synapses. So if we go beyond the individual neurons to the network, there's evidence that there's more uniform synaptic connectivity in regions of the brain affected by seizure. So this is a really exciting hypothesis for us because we think it can sort of unify potentially a lot of different causes of epilepsy that have sort of been thought of their own separate things, but might be able to all be recontextualized as homogenizing forces. So this is something that we're actively pursuing. Homera is in the lab almost 24/7. I don't know how she does it, characterizing these neurons. And we have a bunch of sort of cool directions of computational research that are trying to see what the effects of these different heterogeneities are on networks.